Again, we are in the first perspective of psychology, that is a biological perspective. Here in our first topic, we will go into the details of our nervous system. Some of you may studied about your nervous system, but here we will study it again. And those who have not yet studied about human nervous system, so you must first go to YouTube and first study about the nervous system of human body related to biology. That will better if you will do that, but if not possible, then anyway I'm going to introduce you with the nervous system. And the basic unit of our nervous system is neurons. It was we will be studying about neurons and the transmission of signals from our entire body part to our brain and our spinal cord in the nervous system. So this is a neuron, which is the building block of, which may be called as the building block of nervous system. Actually, it is the unit of nervous system. All the nerve impulses are transmitted through these small nerve cells. These are called small nerve cells. And if I draw a diagram of neurons, it will look like this. This may be regarded as a simple diagram of neurons, but this is not the actual way in which it is drawn. This is for just for your basic understanding. This is a nucleus. This is actually only one cell which I have represented here. There are some parts of this neuron. Okay. So this part is known as dendrite, and this is the nucleus of this cell. Here we may get some cytoplasm, and this is the axon from where the signals are transferred in electrical form. This is known as Schwann cells. This is known as myelin sheath, which may be generated by glial cells, which we will study further. And these are known as synaptic terminals or synaptic knobs. Signals are received by these dendrites, and then they are transferred to the body of this nerve, and then these signals are transferred from within the axon to the other neuron, which leads it to the brain or spinal cord. So We have to understand basically about the transfer of signals, that how it works, its function, its actions, etc. So here the same thing is given in this. This is our dendritic part. There are synapses. There are soma or cell body. This entire part may be called as soma. Axons. These are the axons, and axon is covered by a sheath known as a myelin sheath, and here it has shown some type of action potential about which we will discuss further. Okay. There is something about which we should know more, that is myelin sheath. Uh, this entire cell is surrounded by a jelly-like substance which works as a shock absorber for these neurons. This jelly-like substance will work as a protection to these nerves, so that they cannot be damaged easily, because they are very soft and they are directly related to our brain. So if any damage has happened with these nerves, this might lead to a serious problem. That's why they are protected. There is some a special type of cell known as glial cells. Sorry, glial cells around this neuron. Especially glial cells are around the axons, but they are also present all around the nerves. Now, when the discovery of glial cells took place, we came to know about some of the functions of glial cells, that is getting nutrients for the neurons, cleaning up remains of neurons that have died, communicating with the neurons and other glial cells, and insulating the axons of some other neurons. These were the some of the functions which were discovered earlier, but still the functions of glial cells are being discovered, and it is in its mode of discovery. Hence, there are many new features, new functions are coming in front of us day by day. Glial cells, as per the new discovery in 2005 approx, glial cells have been also said to have some properties of stem cells. What are these stem cells? Stem cells are totipotent. Now, what this totipotent means? Totipotent means a type of cell which has the ability to form the entire body of any organism. Like when we were in our ovaries, in the ovaries, our life was started with only and our life was started with only one cell, and that one cell has grown up so that we are seeing our body right now in this form. So these are the totipotent cells which have the capability to form every part of our body. So glial cells are said to have this property, and due to the effect of these glial cells, we see that some neurons are when some neurons are damaged due to these glial cells, these neurons are recovered to their positions after some times. Talking to the types of glial cells, there are two types of glial cells. One is oligodendrocytes, oligo, oligodendrocytes and Schwann cells. Uh, Oligodendrocytes are found in the neurons of brain, and Schwann cells are found in the neurons of our peripheral nervous system, that is excluding brain and our spinal cord. So oligodendrocytes will be found in our spinal cord and also in our brain, but Schwann cells will found only in the other parts of our body. What happens? What is the difference between both of them? This Schwann cell and this oligodendrocytes both releases myelin sheath. Both forms myelin sheath. Myelin, and this also produces myelin. Now, this myelin insulates the axon of any neuron. Remember, this myelin insulates the axon of the neurons. But when Schwann cells produce the myelin, it forms a tunnel, and within this tunnel, this axon of the neuron passes. And when talking about the myelin sheath produced by oligodendrocytes, it doesn't form such type of tunnel. It only protects as an insulating layer. So when the neuron of any peripheral part of the nervous system, that is part other than spinal cord and brain, gets damaged, it gets restored in this tunnel. Suppose it is damaged from here, and after some times, since there is no external interference, this entire neuron will get repaired auto automatically. But there is a problem with the myelin which is produced from oligodendrocytes. The problem is they don't form such type of tunnel, and due to this, when the neurons of the brain or spinal cord are damaged, there is a most likely chance that it has been damaged permanently, and it cannot be corrected now. So if we can say that the neurons of brain and our spinal cord are highly sensitive to the external factors. Okay, nothing in this. Let's come to our important part, that is creating the message within the neuron, the neural impulse. What is the neural impulse? What is diffusion? What is resting potential? And what is action potential? All will be discussed here. Look it very carefully. Suppose this is one thing. In this figure, the impulse of any stimulus will start from here to here in this direction. That is, the impulse starts from the dendrites, and then it is sent to the synaptic knob, towards the synaptic knob, and then after the synaptic knob, it gets absorbed by the another neuron. Now we were here. Suppose this is our neuron, and this there is our myelin sheath. Where the myelin sheath will be present, the transmission of signal will be very fast due to presence of several type of ions. But where myelin sheath will not be present, the transmission of neurons will be slow. That is transmission of signals. So transmission of signals in our brain will be slow as compared to our other parts of body, that is in peripheral nervous system. What happens? The outer part of this contains positive ions. Let me remove this myelin sheath. This is the axon, and this outer part will contain mostly of positive ions along with some negative ions. Here the positive ions will be sodium, that is Na plus, and some negative ions will also be present in the outer part of the axon. These will be chloride ions, but they will not be in majority. That's why the net potential will be positive. And here it will be negative ion inside the axon. This negative ion will be present in form of potassium ions, and there are some ions which are potassium ions, but they are in a minority. There are some protein negative ions. I am saying protein negative ions. They are big in size as well as great in numbers. Hence, net potential inside the axon is negative, because the number of potassium ions inside the cell, inside the axon, is less, and number of protein negative ions inside the cell is greater, and its size is also greater. Hence, inside the cell the net potential will be negative, and outside the cell the net potential will be in positive. Now, if you have read something, you have a small basic knowledge of physics, you can guess this, that these negative ions will try to enter into the Schwann cells because of the potential difference, and these positive ions will try to enter into the Schwann cell, into the axon, because of the potential difference, and these negative ions will try to move out of the axon due to the potential difference. And this process is known as diffusion, because it is happening because of the difference in the concentration between concentration inside the axon and outside the axon, and this will happen till a balance is achieved. Whatever I told is the situation. This situation happens when the neuron is in its resting position, that is, no impulse, no transmission of signal is taking place inside the axon. So this is a resting potential, in which positive ions are out of the axon and negative ions are inside the axon. And here comes the axon potential. Suppose this is the situation, and now the some of the impulse signal triggers into this axon. What will happen now? Remember, I'm drawing again an axon. There are some gates into this axon. From these gates, positive ion can enter into the Schwann cell, but negative ions cannot get outside this Schwann cell because outside this axon, because the negative ions inside the cell, that is protein negative ions size is greater as compared to the pass available in these gates. But sodium ions are smaller in size and they can pass in, they can move into the axons. But these gates are not open to these sodium ions during the resting phase. These gates are open only when there is some impulse. When some impulse is triggered, then what happens? These gates are opened for a very short time, in thousands of milliseconds. Suppose this is negative charge. Let me make it slightly bigger. There are positive charges outside the axon and there are negative charges inside the axon, net negative charge inside the axon. Now what happens when impulse is triggered? These gates present in the wall of axon gets opened, and due to this these positive ions get into this axon. This will also get into this axon, and what happens when these ions will enter into the axon? What happens is the net potential net charge on the net charge inside the axon becomes positive. This has become positive, and in the same way this charge will flow further. Now this section of the axon will be in positive, this section will be in positive, but all the gates are not opened simultaneously. They are opened one by one. When this gate will open, this positive ion will enter into the axon and it will move further. As it passes further, as this positive charge passes further, this gate immediately closes so that no other positive charge can come into the axon. And meanwhile, some positive charges inside the axon move outside. Which one? That is potassium positive charges, because potassium size of potassium positive charge is small, so they get they went outside the they move outside the axon. So gates were closed, potassium ions were moved outside of the axon. Now what happens to these potassium charges, potassium positive charges which have entered into the axon? Suppose this is our axon potential. Here we have got some positive charges and the signal has got over, I mean it has been transported back to the transferred to the next neuron. Now these positive charges will be thrown out of the axon by its cell wall, because its cell wall is actually inside the cell wall there is a plasma membrane which is semi permeable in nature. It decides which molecules will come inside the cell and which will go outside the cell. So due to this plasma membrane, these positive ions, positive sodium ions, will be kicked out of the axon, and during that time the sodium ion, potassium ions which had went potassium ions which had went outside the axon, they will come back to their original position. Hence, as soon as the signal is transferred from particular point of the axon, the resting potential will remain again. It will reverse back to the situation. This is known as action potential. Action potential is generated only when some signal or some stimulus is received by the neurons. To sum up, this is actually what happens during. I have told all these things about this, and this is the graph and the level of potential or electric impulse during the transmission of action potential. We are not going into the detail of neurotransmitters in this class, but let me sum up what I have told. During the resting phase, there are negative ions, net negative charge inside the axon, and net positive charge outside the axon. Negative charge is mainly composed of protein negative ions having some potassium positive ions also, whereas net positive charge outside the axon is mainly composed of sodium positive ions and some chloride negative ions. This is the resting situation. As soon as the impulse gets started, the situation gets reversed, and now the sodium ions will enter into the axon, and at the same time some potassium ions will get outside the axon to maintain the balance, that is homeostasis. Now when from that point the signals are transferred to other point, situation becomes again same. The plasma membrane inside the cell will kick out the sodium ions, and at the same time potassium ions will again come inside the axon, and this phenomena will be continued till the impulse is reached at the synaptic knob or synaptic terminal of the neuron. After it has been reached to the synaptic terminal, what happens will be we will be covering in our next class, and next class will be all about neurotransmitters. It is very important with respect to biological perspective, that is neurotransmitters. So that's it.